Hi everyone, thank you for joining our video on BioMeld Project. BioMeld is EU-funded Horizon project with focus on modular framework in designing and the production of biohybrid machines. I'm joined today with our esteemed professor, Dr. Igor Balazs, with whom we will explore the deep tech concept of BioMeld Project. Igor, thank you so much for allocating your time to discuss with us. Please tell us a few words about yourself and your current research focus.
Thanks, Ariana, for this nice introduction, and thanks for having me here. My background is quite diverse. I started as a biologist, but then I shifted my focus more towards physics of complex systems and application of modeling and AI in understanding those complex systems. And during my work, I mostly focused on medical application of those skills, especially in medical oncology, taking together both research and clinical side of the question, where each of those raise a separate set of issues that we need to handle together. And that's quite challenging, but also exciting thing to do.
Then please tell us a little bit more about the BioMeld project concept. What is the modular framework and how it works in biohybrid machine setting?
Yeah, thanks. So basically, at its core, BioMeld combines two things, which you mentioned that are in the title of the project. So we have modular framework and biohybrid machines. Biohybrid machines are devices where you combine living cells and artificial engineered materials. Basically, you have living tissue, in our case, muscle tissue, that acts as actuators in the proof of concept we are developing in the project, that are merged together with the basic structure of the catheter, with electronics and sensors, and blend into what we call biohybrid machine. The other aspect is modular framework, and it covers how we basically approach building those things. Right now, in usual practice, different research groups that are in the field of biohybrid machines are mostly working each time from scratch, basically relying on their intuition, their background knowledge, and trial and error approach that emerge from their labs know-how. In our project, what we want to build is a sort of a shared toolkits that will help different laboratories in this field to basically avoid trial and error process and have a clearly defined pipeline from the ideation phase up to manufacturing of a prototype that will help them in the entire process and also alleviate possibility of sharing data and knowledge generated in each of those lab-based smaller projects they have. So what we want to demonstrate is the whole pipeline, how it works, in the use case of building a medical catheter, biohybrid catheter.
What is the difference between these two concepts? Hybrid machines and also nanorobots, as through some of papers that are published on BioMeld project, I had the opportunity to face different terminologies, and please give us your insight to better understand the differences and similarities between these two concepts.
Yeah, thanks. Yeah, really good question, because in usual media coverage, things get blended and they do not follow a strict distinction between different attractively sounding terms. So basically, in biohybrid machines, as I said, we are using living biological materials, so living cells. And they are those tissue samples and tissue actuators are at the level of millimeters, and they are combined with those synthetically engineered components. Nanorobots, on the other hand, are different concept, and the key difference is basically a spatial scale and composition. With nanorobots, you're dealing with nanometer scale, as their name says, which is 1,000 times smaller than human hair. And again, much, 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 much smaller than the spatial scales we are operating on. Also, with nanorobots, you usually talk about either molecular machines or completely engineered structures that need to do some work in subcellular domains. So basically, these are two different domains. Both are kind of futuristic, but I understand that sometimes in popular science writing, they get merged together and they can raise confusion. So that's why I think it is quite important to separate them too.
I would like to learn more about biointelligent design and BioMeld's self-monitoring and self-controlling manufacturing pipeline and how it differs from traditional biomedical manufacturing.
So basically, biointelligent design, way we use it, means two things merged together. First, we use a different set of AI methods, particularly evolutionary algorithms, where candidate designs are evolved through digital generation, uh, multiple digital generations, and that's in sharp contrast to hand-designed devices. Second thing is that we also included a language-based model that lets a designer describe in just a plain language what they need, and the system will be able to translate those relatively free-form narrative explanation of what is in designer's head into a set of technical specifications. If you allow me, I will just share briefly a set, a scheme basically of how we are implementing that. So basically, as I said, the idea is to start from a natural language input requirement in a rather free form where you can just say, I want to design a biohybrid machine for this and that. And the model will then understand what are the basic requirements of that device, where it should be used, and the output will be a set of specifications that you need to take care of during the design of the entire device. We built that first step by first precisely defined or making a defined ontology of all the terms. We developed a set of templates and then developed a LLM-based semantic system that should understand those relatively free-form inputs. As a next step, then goes more precise ideation, where evolutionary algorithms are involved. They start with a relatively vague idea of what should be developed. If it is a catheter, then it should be, of course, small, elongated, flexible, and evolution algorithms work through those requirements and offer to a designer a smaller set of possible candidates that he or she can work further together. Of course, these are just a basic morphological sketches of how it should work. And then the next step is more precise set of simulations that take into account different levels of physical reality in order to further down make a much more narrower space of possible design candidates that then only go to a testing and validation phase in the real lab. As you can see, at each step, we have a clearly defined human decision points. So when you said self-monitoring and self-controlling pipeline, it's more like human in the loop pipeline because it is important to include human reasoning at each of the major decision steps. And within those steps, the entire pipeline work mostly in self-controlling way. So it is a kind of combination of self-control with checkpoints where human decision is made.
So BioMeld Project device is basically a drug delivery catheter intended to enable access to hard to reach areas in human body. So what are the differences between some available drug delivery catheters on the market and future BioMeld product when we compared the devices with the same intended purpose, for example, in oncology?
Yeah, thanks. So before going to straight answer to your question, I would just like to emphasize that yes, reaching hard to reach areas is our long-term vision for this technology. And we are at the moment far from that. What we produced in the project is a proof of concept device, and which is tested in a lab, produced in the lab, with the focus on steering mechanism, which is biohybrid. So I just want to make that part clear that this is a vision. Since it is a biohybrid device and the entire field is in its very early stages, we mostly focused on producing a muscle actuator and integrating it with the rest of the catheter, with sensors, with electrical current wires. So that's part I wanted to make perfectly clear. So drug delivery payload is at the moment just a future development step, not something we have yet validated or produced. But back to your question. The main difference is actually the actuation method. Current steerable catheters typically use pull wires, shape memory alloys, or external magnetic field to bend the tip when navigating the maze of blood vessels. In our approach, we are using living muscle tissues as the actuator, and it is on the fundamental side is completely different physical principle. Muscle tissues are soft, biologically compliant, and in principle, and that's how the entire idea emerged, they could allow for more gentle and finer control of movement compared to rigid mechanical components. And I think it matters most when navigating those delicate blood vessels in hard to reach areas. So our proof of concept is a flexible biohybrid catheter. And as you can see in this slide, it is composed of different things. It has as a basis flexible platform with integrated sensors and with integrated electrodes. And then we have an actuator, which is built of actual skeletal muscle tissues. But since they are living tissues, that they need to be protected. So on top of that, we also have a closed chamber with the flow of a medium that will feed those cells so they can survive as long as possible. So as you can see, that the entire structure is rather complex because you have a lot of moving parts that needs to be precisely integrated together.
When we talk about the paradigm shift that BioMeld project will bring us, what more precisely can we expect of the impacts in the medical technology area that BioMeld project will bring us?
Thanks. So as in at the beginning of our talk, when we discussed the entire concept of the project, so two things are blended together here, and both are kind of paradigm shift in their respective fields. So the first one is methodological. Before a systematic computational framework like this, building a biohybrid machine was, as I already mentioned, kind of an artisanal process. Every lab did it on their own, in their own way, taking inspiration from know-how that they already have. The idea here is to make a paradigm shift in the methodological sense that will enable more structured and better shaped design and manufacturing pipeline of the entire process. And my vision is to move that field from individual craftsmanship toward shared, systematic, engineered practice. That's one part. The broader shift is about biohybrid devices itself and what they represent for the medical technologies generally. Usual way of doing this for implantable devices and minimally invasive devices is to use basically rigid or semi-rigid synthetic materials, whether metals, polymers, or electronics. And it gives us precision, which is, I guess, highly regarded in the regulatory space, and durability also. But the cost that is incurred by such approaches that synthetic actuators are typically stiffer than biological tissue. They are not able to self-repair, and they can trigger immune response. So you need to be very careful about that part. Living tissue, on the other hand, are inherently soft, are biocompatible, and at least in principle, are capable for adapting and self-maintaining in ways no synthetic materials can do. So that's our long-term vision on the side of biohybrid machines, to try to move the field into the direction of self-adjusting materials that can properly react and adapt to a patient body and situation where they are in.
Thank you. Thank you so much. We touched based drug delivery application and purpose of one of the aspects of potential applications of biohybrid machines. but we probably, I mean, you probably estimate more applications, more different applications. So yeah, please give us your,
Okay,
your
yeah,
opinion
great.
on
Thanks.
this.
Thanks. So now we are entering wild dreams, which I like. So basically, what is the narrower focus of this project is to move towards minimally invasive surgical tools that needs very fine, gentle movements and things I already explained. Uh, However, the ability of soft adaptable devices to navigate and sense inside the body opens up a huge variety of possible applications because don't forget that our entire prototype of catheter also include sensors attached to it. So by changing sensors, basically you can probe different parts of the body, parts of the tissue, for a variety of biomarkers that are possibly presented there. So it can be used in a wide scope of endoscopic tools for either sensing or intervention in the body. Also, just a brief mention that we also started looking into possible applications outside medical applications entirely, because the same principles and the same computational design framework is relevant to industrial soft robotics for things like inspection robots that are able to move through confined or very irregular spaces. So the scope of possible applications is quite wide. Of course, each of them will require a lot of effort, a lot of funding. But what we intend to do with this project is basically to build a basic building blocks to move forward.
Thank you. Thank you. We touched base again through all these responses, the purpose of BioMeld project and similar projects, and we are here talking basically about next gen precision medicines. And I would like to hear more from your perspective on patient outcomes, what we expect from what are our expectations, and how we see in a long run improvement in patient outcomes.
Yeah, okay. So I think, well, my answer can be quite short. Two main things, lower doses and fewer side effects. And maybe I can stop here because lower doses can be achieved with better and gentle and more precise reaching those areas where you do not need to inject higher doses of treatment molecules. If you are in the close vicinity of something, then you can be quite precise with dosing. With fewer side effects, we touch upon two different things. One emerge from lower doses, so you do not need a chemical compound to enter in large quantities systematic circulations, because in oncology, one of the main limiting factors in application of chemotherapy is basically their systemic circulation that harms other organs. But also fewer side effects can stem from more precise and more gentle navigation of endoscopic devices, where the probability of damaging host tissue is much lower. So, as I said, I think it goes into those two things: smaller doses and less side effects.
Thank you. And when we talk about BioMeld project and its next steps, what do you see as the next steps and next stage for the BioMeld product?
Product aside, we developed a lot of data and knowledge that can be useful for other domains outside of the catheter application itself. So basically, we have two patent applications for different aspects of actuator mechanisms, mechanism that is implemented here, and for a chamber that needs to be integrated around muscle actuator. We also produced and put it in open use ontology for LLM-based system. We produced and again put it into an open space simulation data. So we are making a lot of different building blocks that should help us and other groups in pursuing further with different aspects of biohybrid machines. For the catheter itself, since it is intended as a medical device, then as you very well know, the journey toward that is quite long, given all the regulatory requirements. So as immediate next steps, I see further steps in integration and miniaturization of the entire device. It's testing in more challenging environment, in close collaboration with regulatory experts, who will then become basically a major guide toward market, because we need to know what exactly are the main pain points in the future development and in ultimately obtaining a regulatory approval for that.
Fantastic. And here we see some opportunities for further development of Project BioMeld project and opportunities to even deep dive through the healthcare aspect, medical aspect of BioMeld project. Thank you so much, Igor, for allocating your time and exploring with us, demonstrating to us all these critical aspects in such a critical area such as next-gen precision medicines in oncology. Thank you so much.
Thanks a lot, Irena. It was a real pleasure.